Okay. Hello. I hope that you guys are all doing well. Um, Remember that we're going to have the T cell development problem set to on Monday next week, not on Friday, since I'm a little bit behind. The PRR assignment is still due Friday of next week. Both of them are posted on Moodle. And I am in process of grading the problem set that you gave me on Monday. Today I'm going to talk about T cell development. I just wrote a little table on the board about B cell development that we're going to use for compare and contrast a little bit later today. So we'll get to that. I just wanted to have it up on the board before I got started. So if you remember what I was mentioning last time, one of the things to be aware of is that T cell development and selection processes are all happening in the thymus. So everything that we're thinking about today is going on in the thymus. And T cells are developing from the common lymphoid progenitor. So that was something that came from the original hematopoietic stem cell. That stem cell could decide either to become a myeloid cell or a lymphoid cell. And then that cell makes a decision to either become a B cell or to become a T cell. And once it sort of makes the decision that it's going to start doing T cell development and not B cell development, that cell will move from the bone marrow to the thymus and start some of the processes that we are going to see today. I left off last time talking a little bit about the thymus, specifically mentioning some details of the structure of the thymus. And so you can see that the thymus contains two areas known as the medulla and the cortex. You can see that they stain differently in this histological stain. And that difference is largely due to the density of cells in the two areas. You can see that the cortex is much more densely populated than is the medulla. And you can see that, in fact, there are a few other areas that can be labeled here. So you can notice that. You can even see here, this area is more densely filled with these purple cells than is this area. So the cortex and the medulla are pretty easily distinguishable. In this image from your textbook, the developing T cells are in blue. And you can notice that there are also a whole lot of other cells shown in the thymus that are not blue developing T cells. And those cells are cells that we are generally going to call thymic stromal cells. Sometimes we'll think about them as thymic epithelial cells. You can think about them as being the cells that are part of the structure of the thymus. And we need to remember that those cells play a very important role in T cell development. So sometimes we need to think about what those epithelial cells or structure cells or stromal cells are doing, in addition to what our developing T cells are doing. As we go through the stages of T cell development, one thing that you will see is that our T cells kind of move to different locations for different steps of the T cell development. So we're going to actually see the cell moving between the cortex and the medulla for different parts of the process that we see today. And so know that while we're in the thymus, the specific area within the thymus might vary depending on which step we are at. One of the reasons why it's so important that I point out the structural cells or the stromal cells in the thymus is because some of the experiments that have been done that have helped us understand T cell development involve some different kinds of manipulations we can do with different mice. So one manipulation we might do is we might take a mouse that doesn't have a thymus, and I'll say more about a mouse that doesn't have a thymus in a few slides, and we can transplant in a thymus. And when we're doing that transplant, the thing that's really key when we do that transplant is that we're transplanting in like a new thymus structure. It's really, at that point, it's about the stromal cells, the epithelial structure cells. So just to give you an idea here, this mouse is of the yellow genetic background. And we might transplant in a thymus of the green genetic background. So the thymus and the rest of the mouse have different genetics, and we can then see what effect does that have on T cell development. So sometimes we do that, and really the only thing in this mouse that is green is the structure cells of the thymus. Everything else, including the developing T cells, are yellow because those developing T cells came from this mouse's bone marrow and then went into the thymus to do their thing in the thymus. We could also do things where we might take bone marrow and put it in a mouse that doesn't have its own bone marrow, or in a mouse that we've done radiation on to get rid of the bone marrow that it has, and look at what happens again if we do things like mismatch the T cells and the genetics of the thymus. So here we've got a green mouse, and we put in yellow bone marrow, and so again there's this little bit of a mismatch. Both of these types of experiments have told us one really important thing. And this is why I mention those cells of the structure cells of the thymus. And again, those structure cells of the thymus, if we say do radiation, they don't get wiped out. The developing T cells, the bone marrow gets wiped out, but the structure stays the same, which is what happened in this mouse. What we have found is that, as the top of the slide says, T cells are restricted by the MHC of the thymus. What that means is that when a T cell develops, it has to learn which MHC to recognize. It has to learn which MHC is mine, which MHC is self that I'm supposed to recognize, right? It's got to figure out what is the correct MHC to be recognizing. And it learns that from the thymus. It actually learns that from its interactions with the structural cells of the thymus. So in this experiment, we have bone marrow coming from an orange mouse. And we can transplant it into either a yellow recipient who has a yellow thymus or a blue recipient who has a blue thymus. And if we do those experiments and we look at these T cells that grew up from this bone marrow, if I had just left them in the orange mouse, those T cells would grow up and they would think that orange MHC is self. But if I put that orange bone marrow and those orange T cells into a yellow mouse, those T cells develop and learn that yellow MHC is self. And they only react with yellow MHC, and they don't react with blue MHC. If I put those same orange developing T cells into a blue mouse that has a blue thymus, particularly the blue structure cells of the thymus, those T cells grow up learning that blue is self. And they only respond to antigens presented on blue MHC, not on yellow. So the structure cells of the thymus, the stroma or the epithelial cells, are particularly important for helping the developing T cells know what MHC is self. And as a result, T cells are always restricted by the MHC of the thymus. So if we ever do like these mix and match experiments, and we wonder what MHC do the T cells recognize at the end of all of this crazy business? The answer is always whatever MHC the thymus structure has. And so, just as an example, here we had a green mouse and we put in yellow bone marrow. And here you can see there's the thymus right there. So what are the T cells that develop here? What are they going to think of as self? Yeah, Olivia. Green, because they're growing up in a green thymus. But even though they started out being from the yellow mouse and they have the yellow genetic background. Okay, what about in this case? What are the T cells going to think is self? Yeah, Vicky. Green, because it's a green thymus. Even though the whole rest of the mouse is yellow, even though really in this mouse, having T cells that recognize green is going to be kind of pointless because nothing else is green in this mouse. The T cells are going to have learned green is self because they do their development in the thymus. So you want to just be aware that whatever kind of crazy manipulations we do, the outcome is always T cells that are restricted by the MHC of the thymus. And that's because of those interactions with the structural cells of the thymus. There is one other thing for you to know. I'm actually going to do the next two slides in reverse order. I mentioned to you before this idea about mouse strains, that once upon a time there were some people who observed mice that had weird phenotypes, and they said, that's weird. And they started mating the mice with the weird phenotypes together to see if they could get it to like breed true. And that's how we got a lot of our inbred mouse strains. So sometimes they would just observe a mouse that had like a weird characteristic. Now we do genetically engineer them. But originally, all the first big mouse strains were observed based on just, oh my gosh, I found this mouse. It looks funny. One of the big mouse strains that people observed that looks funny is shown here. And it is known as the nude mouse. And you can see the nude mouse is here. Nude mice, and again, this is a spontaneous mutation that was just, these mice were just discovered, though now we've bred them, and so there's a true breeding nude mouse line. These mice, you can notice, have no hair. They also have no thymus. The hair and the thymus don't really have anything to do with one another. It's just, this was actually a natural line that was found. But we can do a lot of those different types of transplantations that I mentioned to you, where you can transplant in different thymuses and look at all sorts of different things with these mice because they don't have a thymus themselves. And in fact, if you have this mouse just hanging out like this as a nude mouse, it will have no T cells because it has no thymus and no place for T cells to develop. And so it's a very immunodeficient mouse because it has no T cells. Of course, you can do a transplantation. You can transplant a thymus in, and it will be fine, and it will get T cells, and it will live happily ever after. It will not grow hair because, like I said, the two things aren't actually related to each other. Once I had students reading this paper, it's a really important paper, but they use nude mice a lot. And I asked one of the discussion questions was like, why did they use nude mice? And the answer is because nude mice don't have a thymus and don't have T cells. And these students all wrote me these like amazingly long, involved things about, well, because they don't have hair, you can see skin transplants better and things. Yeah, the no hair doesn't matter here. It's really about the no thymus and thus no T cells. Just so you know, there are actually some people who are born without a thymus. It is a condition called DiGeorge syndrome. It's an immunodeficiency in kids. The kids have hair. So first of all, there is no link here. But with DiGeorge syndrome, we have a pretty easy fix, just like we do with the nude mice, which is that we can actually transplant in a thymus. And so if you transplant in a thymus into those kids, then they can do T cell development and everything's totally fine. One thing that we've actually also learned is you don't have to put the thymus in on top of the heart for it to work right. It just has to be somewhere. So in mice, we usually put the thymus in the kidney capsule because it's easy to do a transplant into the kidney capsule. For the kids with DiGeorge syndrome, we usually put it in their thigh, and it all works just fine. That's one thing to know about the thymus. But there's one other thing to know about the thymus, and this there's lots to say here. So it has been observed that as people age, their thymus changes. In this middle picture, you see a thymus, and you can see cortex and medulla really nicely. And that is a thymus from a young individual, and I don't remember the age of the individual there, but that's probably a thymus of like a kid, maybe like a 10-year-old or less. Okay? On this right-hand side is a thymus of an older individual. And you can see much less of that nice cortex and medulla, and you can see all of this other space that's sort of invaded. And in fact, generally that is invasion of fat, with fat cells invading in. This is known as thymic involution. And the thing that is important about this is as one ages and as this happens to the thymus, this reduces the amount of the thymus that is usable for T cell development. Basically, your thymus can't do as much T cell development when it looks like this compared to when it looks like this. And in some ways, it's actually like a surface area of good area stuff. So here, there's only a little bit of good area for T cell development. Here, there's lots. And if you look at this graph on the left, this is showing kind of how much T cell production people have at different ages. And so you can see, in your first year of life, you make a lot of T cells, and really up until like 10, you're really making tons. And then it goes down over time. This is important for a bunch of reasons, but I do want you to also look a little bit closely at those data. One reason why this is important is because, in fact, it thus makes it important that you make good T cells when you're a kid, because your thymus is working the best when you're a kid. If you actually try to do a dissection on a mouse, and you get a mouse that's like older or an older individual, the thymus is like smaller and ickier and like falls apart. When I do a dissection when I have a lab section of this class and we do thymuses, I have to actually control the age of my mice to make sure that there's a thymus that you can actually like get with a pair of forceps. Because if the mouse gets too old, it's just this fatty thing that disintegrates when you try to touch it. So that's one thing to be aware of. If you end up being a physician, or in fact, if you ask a physician, particularly imagine like some super old MD physician, you ask them, what they will tell you, and what is often talked about in med schools, is that the thymus stops working as you get older, or the thymus doesn't do anything in adults. So I want you to notice, yeah, it decreases, but like it doesn't go to zero. And it in a lot of like medical textbooks, they're basically like the thymus is only useful when you're 10 and younger, and it does nothing in adults. So it does decrease. Older people are making fewer T cells, but it's not zero. And so I always get a little feisty when I hear stuff like that. So notice, yeah, it does go down, but it doesn't go down completely. That also has led to some ideas. I'm going to tell you guys this story, and it's a key story. It's an important story. And every time I tell it every year, students are just like horrified and make these awful faces at me. So this has led to a lot of physicians to have this idea the thymus doesn't do anything in people, or the thymus isn't useful in most people. It's kind of like, that's weird. So you might wonder why I'm telling you that, or why that is in some way important. In the lab where I worked as an undergrad, we had this giant freezer full of pieces of people's thymuses. That we did all sorts of interesting studies on thymuses. Sometimes some of those pieces were used for transplantation into DeGeorge patients and things like that. And you might wonder, why did we have all these pieces of people's thymuses? What's the deal with people's thymuses, especially kids' thymuses? And the answer is whenever somebody has cardiac surgery or heart surgery, especially if you're younger, the thymus can actually be like a little bit in front of the heart, and the surgeon will just cut it off because they are afraid it is in the way of their heart surgery and is going to complicate the heart surgery. And so in many cardiac surgeries, the thymus is either entirely removed or partially removed. Part of that is because of this outdated idea that the thymus just doesn't do anything in adults, so why can't we just cut it off? That is nice in that it means that we have thymus sources to study when we want to study the thymus. It's nice because then we have things to give kids with DeGeorge syndrome, but I kind of want to keep mine. Even if I am old and it's not doing as much as it was doing in the past, I'd like to keep it. So in fact, just know that the sort of anatomical location here does end up playing a bit of a role in some important details. And it's still working in all of us. See, none of us are at zero, not even old me. Okay, so now we need to start getting into sort of some more specific details of T cell development in terms of phases and events and all of that kind of stuff. When we talked about B cell development, we use this framework that I've written over here on the board. So we talked about 3 different kinds of B cells. We cared about what was on the surface of those B cells with regard to the B cell receptor, and we cared about what kind of process B cells were doing. And this, of course, is a very sort of broad overview. If I, again, were to go, and like I said, note the words broad overview, if I was going to give you a broad overview of T cell development, I want to do a comparison with what you see here, okay? Okay, I'm done. So what you will notice is that in very big picture, very broad overview terms, T cell development has a lot of similarities to B cell development. And basically all I did on my overview was erase wherever it said B for BCR and write a T in its place. And so you can see we can think about different phases of T cell development. We can think about them having different things on their surface and doing different processes. And all of that framework totally works for us thinking about T cells. But T cell immunology can sometimes be slightly annoying. And so we also use some different terms when we talk about T cells. So we don't as often talk about it quite like this. But in the end, we're going to be just putting more things onto this framework that we've already seen. So it's really stuff you've already seen, just with a little bit of additional stuff. And we are really going to be starting from this cell that has decided, I would like to be a T cell, and that has gone to the thymus. When we look at T cells in the thymus, if we just take all the cells out of the thymus, we're really going to get those T cells at different stages of development. So you can imagine if I take cells out of the thymus, I am just getting the developing T cells. Imagine I don't get the structure cells. And kind of that's really is what happens. And I don't know that I can explain why that is, but just go with it. So sometimes I'll talk about thymocytes or cells of the thymus. And thymocytes are all developing T cells. So in fact, I don't have to like gate T cells versus B cells in the thymus. All I got is T cells. It's just 100% developing T cells at different stages of development. I can use my flow cytometer to divide those cells up into some groups. The first thing I will do with my cytometer is I will have CD8 on one axis and CD4 on one axis. So I'll make a plot of CD4 versus CD8 in the thymus. And when I do that, I will get 4 groups of cells. One group of cells will be CD4 negative and CD8 negative. We often refer to those cells as the double negative population. You can notice that the double negative population is the smallest population in the thymus. It's listed here as being about 3%. You can see that we have one population that is CD8 positive and CD4 negative. We refer to those cells as the CD8 single positives. And you can see that those are a little bit more than the double negatives, still not huge, so they're like 5%. You can see that we have one group of cells that are CD4 single positive. So they're CD4 positive, CD8 negative. They're kind of the next largest group at 12%. And then we have this final group that's CD4 positive and CD8 positive. They're called the double positives. And the double positives are the most frequent cells in the thymus. You can see that they are at about 80%. So if I were going to draw this, I would draw it looking something like this. Have a tiny circle for bi-double negatives because they are the least frequent population. Maybe a little bigger for my CD8 single positives. Maybe even a little bigger than that for my CD4s. And real big for my double positives. And no, I'm not going to like get out a millimeter ruler and measure the difference between these two. Really, I want to see this one being the smallest and this one being the biggest. And I'm not going to get super like worried about the other two. They should just kind of be in the middle somewhere. And so when we talk about T cells at different stages of T cell development in the thymus, we will often put them into one of these 4 groups: double negatives, double positives, CD4 single positives, or CD8 single positives. We have also realized that some of these 4 groups are not totally homogeneous groups of cells. And so we can take the double negative cells, once we know they're negative for CD4 and for CD8, and we can look at 2 other proteins on their surface. So we could take these cells, we could kind of double click on them, and then we could look at their expression of 2 other proteins. And so note that this is on the total double negative population here. We've eliminated everybody else before we did this population. We have gated on double negatives and are only looking at double negatives here. And we can look at their expression of CD44 and CD25. You can see that we have a CD44 plus CD25 minus population, double positive, CD25 plus CD44 minus for both. These are labeled as DN1, so double negative number 1, DN2, double negative number 2, DN3, double negative number 3, DN4, double negative number 4. And so you can see kind of the way they go clockwise around the plot. During T cell development, a T cell does the double negatives in that order. So they do number 1, then number 2, then number 3, then number 4. So that's kind of why those numbers matter. And this is just something that people have observed. So oftentimes, even though we're really talking about pro T cells and pre-T cells and immature T cells, we use names like double negative or double positive T cell instead of using these names that you've seen before. If you even remember, when we saw B cell development, sometimes we said that multiple events happen. For example, with our pro cell, we had one time where D to J was happening and one time where V to DJ was happening, and we kind of talked about like early versus late. Or we talked about like the testing and then more rearranging, early versus late kind of stuff. That's basically exactly what we're doing for our distinctions here too. So the cells that you kind of think about that we might think about as a pro T cell are really the first two cells in this process, which are the double negative 1 and double negative 2 population. And so for pro T cells, we really have DN1 and DN2. And we can kind of see that DN1s are sort of the cells that are coming into the thymus, and DN2s are the ones that are kind of starting to do the heavy chain rearrangement. The DN1 is the cell that's entering the thymus. It's entering right at the bridge between the cortex and the medulla. And then it's going to move into the cortex in order to start doing its rearrangement as a DN2. Okay? So that's what's going on with DN1, DN2. It really looks like what you've seen before is a pro cell. So once we have a cell that has done heavy chain rearrangement, in the case of B cells, what do we call you if you finished heavy chain rearrangement and did heavy chain rearrangement successfully? What do we call that B cell? Yeah. So that was a pre-B cell. When it had finished heavy chain, done it successfully. And what did that pre-B cell have on its surface? Heavy chain and surrogate light chain, which we together called the pre-BCR, right? So now here we're seeing cells rearranging heavy chain. The next thing they're going to do is put that heavy chain on the surface of the cell along with a surrogate light chain. This not, of course, as a pre-BCR, but as a pre-TCR. The pre-TCR or the pre-T cell receptor includes the TCR beta heavy chain as well as a surrogate light chain. And the surrogate light chain in the case of T cells is known as PT alpha. Sometimes it's also referred to as pre-T alpha. I actually hear it and see it as both. I don't care if you call it PT alpha or pre-T alpha. It's the same thing. And you can see that at this point, we already have CD3 around to allow for signaling. Just like in the case of the B cell receptor, the T cell receptor can't signal if it doesn't have a heavy chain and a light chain. And so it's a little bit of a question of, like, it can't see antigen. Sorry, it can't see antigen without a heavy chain and a light chain. So you're like, well, then how does this thing signal? and get a signal that it did a good job, so it gets a pat on the head. And what we think happens is that a whole bunch of pre-T cell receptors will kind of bind together with the different beta chains binding to pre-T alpha so that they can signal together. But again, this is actually not something that we totally understand super well. So based on your knowledge of these cells, I guess I should show you the next slide first. The next slide both shows you something important and kind of also gives away some of my question. We're in the pre-T cell phase, but I haven't told you what we're going to do terms-wise with these new T cell terms I've told you about, like DN1 and DN2. What would you guess might be what we get for this pre-T cell? Yeah. DN3. Oh my gosh, you're right. So we're going to be seeing DN3 and we're going to actually see DN4 in the pre-T cell. If you think about this pre-T cell, in fact, you can even see it here. There it's showing us DN3. Once we get that heavy chain on the surface, we're now a DN3. Once you have that heavy chain on your surface, that pre-T cell receptor, what kind of signals do we want should that T cell get? What kind of events should be going on in that T cell's life when we're testing that receptor? What signals or what things do we want to tell the T cell? Yeah, Joel. Okay, so we might want to identify MHC, but we don't have a light chain yet. So we're probably not going to deal with antigens yet. What do we want to do? What kind of signal should we tell that cell just based on its heavy chain? Yeah. If the heavy chain is good, then you should build light chain. Okay, so we want to tell that cell, so we want to say like, hey, you made a good heavy chain. Like you have a protein. You didn't make a frame shift, so you didn't get a whole protein, right? So you want to tell it you made a good protein. We're going to say you should make a light chain. What else should we tell that cell? You made a good heavy chain, so what else do we want the cell to do? Yeah. To survive. Good. You made a heavy chain. Please don't die. Right? So we're going to say, okay, number one, we want you to survive. You did a good job. What else do we want that cell to do? You did a good job making a heavy chain. What would I like you to do? You're right. Emma's right that eventually we're going to make a light chain, but we're going to do some other stuff first. Yeah.
Once it has like the necessary heavy chain, would it stop doing more rearrangements?
So it's going to stop rearranging more heavy chain, right? So because we don't want to get two heavy chains from both chromosomes, we only want to have the one good heavy chain. What's that process called where we stop rearranging the other chromosome? Yeah. Allelic exclusion, exactly. So we're going to do some allelic exclusion of the heavy chain. So we don't make any new heavy chains. One more big thing, well, two more things I think that are on the slide. What other thing might you want to tell that cell to do? As I think about it, there are actually 3 events that happen here. One of them you won't figure out, but the other 2 you could. Yeah, Joel. Proliferate. You want to say, hey, cell, you made a good heavy chain. Make lots of progeny so that they can each make a different light chain that we can pair with that heavy chain. So we want to survive. We want to proliferate. We're going to do allelic exclusion. In order to proliferate, what are we also going to probably need to do? there anything we need to turn off if we're going to proliferate? Yeah. RAG. We're going to turn RAG. We're going to be like, no more RAG, no cutting the DNA while we proliferate. We're also going to turn off surrogate light chain production because we don't need it anymore. We already use the surrogate light chain. So you can see all of these things happening here. We're going to survive in this cell. We're going to shut off surrogate light chain. We're going to do allelic exclusion of beta. We're going to proliferate. We are eventually going to start making light chain, but before we start making light chain, we're going to do one other thing. And that one other thing is we're going to start making CD4 and CD8. And so that's the one that you wouldn't have necessarily figured out. And so what do you think we call the cell? And so you can kind of see all of this is happening in DN3 and DN4. What do you think we call the cell when it starts making CD4 and CD8? Both together? Yeah, double positive. So this cell is now going to also go from double negative towards being a double positive. And so I was always taught that like alpha rearrangement or light chain rearrangement kind of happens like on this arrow. Sort of like while the cell is going from DN4 to double positive, it's doing the light chain it's like actually doing its light chain rearrangement. And so you can see these are those double negative steps here as well. We've got our 4 double negative steps where first we migrate to the thymus, then we've got beta chain rearrangement, then we are going to start doing some of those selection processes in DN3 to DN4, all the way to getting ready to become a double positive. And all of these steps are happening sort of throughout the cortex. The cell is doing all of these double negative steps throughout the cortex. So there are a couple other things I want to point out with regard to this. When I told you about the cortex versus the medulla, what was the big difference that we saw with the cortex versus the medulla? Yeah. The colors. And what led to the difference in colors? The density of cells. So the cortex was darker and it had a higher density of cells, right? Based on what I'm showing you here, can you imagine any reason why the cortex might be very densely populated with cells? Yeah, Emma. Yes, but there's another reason as well. That's where the cells are dividing a lot. So because that's where they're dividing and having all their progeny, it's going to be crowded. I point this out because for the rest of the semester, we're going to have these like dark and light areas in certain organs. Every time, the dark area is where proliferation happens. And it's dark because the cells are making lots of progeny and it gets crowded. So the reason why the cortex is so densely populated is because there is a lot of proliferation happening, is at least part of what's going on here. But in all of this discussion thus far of T-cell development, there is a piece that I have been overlooking, which has to do with when I was just kind of like, heavy chain, heavy chain, heavy chain. Because if you remember last time, we talked about the fact that there are two different kinds of T cell receptors, alpha, beta, and gamma, delta. And so also during these steps, the T cell is making the decision about if it's going to be an alpha, beta, or a gamma, delta. And so the cell is also doing its lineage commitment to become, to decide whether it's going to become alpha, beta, or gamma, delta. So this image shows you that gamma deltas can sort of branch off in the middle here. It shows gamma delta could happen from a DN2 or a DN3. The cell could be like, nope, I'm out. I'm gamma delta now. Don't want to do this alpha beta business anymore. And so you can see the cell could also make the decision to go to gamma delta during this process. And in fact, I was sort of glossing over it, but some of that is mentioned in this table that you've seen before as well. And so the way that this works is shown here. We talk about the T cell as doing rearrangement of heavy chain first, and heavy chain is the sort of most common heavy chain is beta. But in reality, T cells, when they are double negative cells, start rearranging 3 of the 4 chains at the same time. They start trying gamma, delta, and beta all at the same time. If they finish beta first and get like a good beta that doesn't have a frame shift and actually is like a legit beta that can signal, then that sends a signal and it stops all the rearrangement of beta, gamma, and delta. So it allolically excludes all the other ones, it allolically excludes beta, but also gamma and delta. Because it's like, oh, I got a receptor, I'm good. But if during the time where the cell is rearranging beta, it actually finishes a gamma and a delta first and gets a good gamma and a good delta before it gets a good beta, maybe because it makes a frame shift in its first try at beta or something. If these two happen to finish first, we get a signal, and that allolically excludes and stops further gamma and delta and beta selection. And so the cell is actually going to do beta, gamma, and delta at the same time. That is really 2 heavy chains and 1 light chain. It's going to be doing all of them at the same time. If beta wins, you get a good functional beta chain first, you become an alpha beta. If gamma plus delta together wins, then you get a gamma delta cell. And so all of these are happening together. You can also kind of see this happening here. So here is our cell. It's sort of trying to do rearrangement of beta, gamma, and delta here as a DN3. You can see that kind of happening over here as well. Sort of we're at DN2, DN3. And then that cell could have gamma and delta win and become a gamma delta, or it can have beta win and it could become an alpha beta. And so you can see kind of that's where this split happens. Some of this is based on, and so one also other thing to note is that there are some places where the cell can kind of go backwards and re-choose. All of this is happening based on the details of the signals that are happening downstream of the receptor. So we think that the cell gets slightly different signals from gamma delta versus the beta plus pre-T cell receptor. And so that changes kind of what's going to happen to that cell as it finishes growing up, what functions it's going to learn how to do, all of those things based on different signals happening. And so again, you can see here the cell is choosing gamma delta or alpha beta. Notice that here the cell is kind of picked beta, but it could change its mind later on and pick gamma delta. And the cell is finally committed when it makes alpha. If the cell gets to the point of doing light chain rearrangement and making an alpha chain, remember that the alpha chain deletes the delta locus. So now the delta locus gets thrown away if you're doing this, and we're out of luck. There's no more chances. You can't go back. You have made your decision. And so the gamma delta cell, when it eventually gets a bunch of signals, will make its decision and stop rearranging. Or the alpha beta cell will make alpha, and that will mean it can't make delta anymore, so that's made its decision. And so you can see that also in the cortex, the cell could decide to become a gamma delta cell or can become an alpha beta cell. We're going to see that cell is now a double positive. It's going to have a heavy and a light chain, and we're really going to be talking about the alpha betas from now on. And we're going to have TCR alpha made. TCR alpha is not perfect allelic exclusion. About 20% of alpha beta T cells make more than one alpha chain. And we don't totally know the details of that. Honestly, the biggest reason I know it is because it messes up certain types of experiments. Because T cells can sort of get around what you're trying to do to them by not being perfect allelic exclusion. So I will just point that out, but that's sort of not super key. And then the next thing that we've got to do, just like we did in B cells, is we have to do our central tolerance testing. What was central tolerance testing about in B cells? Yeah, Emma. So it's about testing all of the developing B cells to make sure that they don't recognize self-antigen, and if they do recognize self-antigen, to do something about it before we let that B cell go into the periphery, right? That is also a big part of T cell central tolerance testing. But T cell testing has one other part to it, that in addition to checking for self-reactivity. And this is actually something that Joelle mentioned earlier. So there's one other thing we've got to test our T cells for. We have to test, are they self-reactive? And, going to give us autoimmunity. That'd be bad. But what else do we have to test our T cell for? MHC. We have to make sure the T cell sees MHC. If the T cell sees something other than MHC, because remember, we're doing this totally random receptor recognition, or receptor rearrangement. Maybe we could get a V, D, and a J and some P nucleotides and N's that don't recognize MHC. So we got to make sure we recognize our personal MHC, or at least whatever MHC we see in the thymus. And we have to make sure we're not self-reactive in the T cell, or in the thymus. And so now we're going to see this part of the process happening. And all of this is going to be happening in our double positive cell. What I want you to notice here is that a lot of cells are dying during this testing. A lot of cells are going to fail, either because they bind self-antigen or they don't bind MHC. If you remember, I told you about that book last time and people talk about T cells going to college in the thymus, and I said this book talks about the thymus as murder university, because so many cells die there. If you look at our little plot here of single positives, double positives, and double negatives, our cells started out here in this small population, and then they went like this, right? They were double negatives, and then they became double positives. There's not very many double negatives, and there's a lot of double positives. How do you think we got from a small number to a large number? Here. in the cortex? Remember the densely packed one? Yeah. Proliferation. A lot of this is about proliferation having happened, so we get a lot of cells. Where eventually the double positives are then going to become one or the other single positive. And notice that the single positives are way smaller populations. And a lot of these events are going to be happening in the medulla. The medulla, if you remember, is the area that is not as densely packed. So one reason why the cortex is super dense is because of proliferation. After the cells are done in the cortex, they're going to go to the medulla. And some of this testing is going to be happening in the medulla. I just white boxed it out for right now. Why might you guess the medulla also doesn't have as many cells in it, is not as packed? Why is the medulla kind of not so packed in addition? Yeah, Vicky. Yeah, a lot of the cells that make it there die. So it's going to be less crowded. And so we're going to be seeing a lot of cells dying during this testing phase of T cell development. And one way of thinking about this is kind of thinking about it as shown here, that you can kind of think about our T cell getting two different kinds of signals. There are pros and cons to drawing it this way. There are some reasons I like it that this book drew it this way. There are some reasons why I don't like it. What I want you to notice is that you are going to be thinking about a signal called positive selection and a signal called negative selection. Okay? And we'll get to differentiating what exactly is going on in between those. But just from the names, positive selection and negative selection, would you like to have positive selection happen to you? Does that sound like something you would like to experience? Yes, positive selection sounds great. Positive selection is a good thing to have happen to you. Would you like to have negative selection happen to you? Negative selection is bad. So we're sort of going to be thinking, does the cell do positive selection? Yes or no? And does the cell do negative selection? Yes or no? And you can imagine one of them you kind of want, and one of them you don't want very much. When immunologists think about positive and negative selection, we often describe and think about it with a model known as the Goldilocks model. The Goldilocks model, in reality, has 4 outcomes. I am not going to totally tell you about one of the outcomes now. I will tell you about that outcome later. So what I want you to know is that there's one outcome where I just kind of have a box here. And we're going to talk about that one more later. So know that it's there, yeah, but I'm kind of ignoring it for right now. It's also here, and I'm kind of ignoring some of it for right now. Okay? And basically, the way that this works is based on how much signal the T cell gets in the thymus. So let's imagine, and one other way you can think about this is that T cells are real needy. Okay? Let's imagine the T cell gets no signal in the thymus. First of all, let's think for a second. What kind of things are present in the thymus that could allow that the T cell receptor could bind to? What is there in the thymus for a T cell receptor to bind to and maybe get a signal from? Yeah, Grace? All those structural thymus cells, absolutely. And what do you think those structural thymus cells have on their surface that's going to be helpful for the T cell receptor? What is going to get bound by what on the structural cells of the thymus will be bound by the T cell receptor? Yeah. MHC. All of those structural cells are nucleated cells. So they all have to have MHC class 1. When we talked about professional antigen presenting cells and cells that had MHC class 2, I said, well, it's the professional antigen presenting cell and this one other kind of cell that's a structure cell of the thymus. So there are cells in the thymus. Everybody's got MHC class 2, and there's a special set that has everybody has MHC class 1. There's a special set that has MHC class 2. And the things that are being presented in the thymus are really MHC and self proteins. Okay? You should imagine there's no pathogen in the thymus. There are situations where there's a pathogen in the thymus and then everything gets all wacky. But you can imagine no pathogen in the thymus. So it's just MHC class 1, MHC class 2, and self antigens. What does it mean if the T cell binds to absolutely nothing and gets no signal at that point while it's in the thymus? What does it mean that cell binds? Probably nothing. It doesn't bind MHC. If it cannot get a signal from the thymus with like all of the thymus full of MHC, if it gets no signal, that cell does not bind MHC. And so if that cell has very, very low or no affinity, so very low or no binding, that cell doesn't even bind to MHC. Is that cell useful to us? No. In fact, sometimes we refer to that cell as useless. Do we want to keep that cell? No. We don't. It turns out we don't actually kill that cell. We just never give it a signal and tell it to survive. And so this cell ends up dying by what is known as death by neglect. And you can see that here. So we've got our cell that's going to undergo death by neglect. We also sometimes will say that that cell failed positive selection. It got no pats on the head. So it failed and it died. So it failed positive selection. What if we have a cell that gets a really, 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 really strong signal in the thymus? Well, the only thing it can get a strong signal from is self-peptide. So that means it's self-reactive, right? So that one is like high. If we get a really high signal in the thymus, we need to kill that cell because it's going to give us autoimmunity. And so we're going to negatively select that cell and we're going to kill it. Now notice the cells that died by death by neglect is like 90+% of the cells. And a decent percent are also getting killed by negative selection. Sometimes we will say neglect the useless, kill the harmful, because this could be a harmful cell. But if a cell gets a intermediate amount of signal, a little bit of signal, or as Goldilocks might say, a just right amount of signal, then that cell probably is binding to MHCs a little bit, but maybe isn't binding the peptides very well. So it's just getting a sort of lowish intermediate signal. That cell will be positively selected. That cell is useful. And we keep the useful cells, and those cells actually will leave the thymus and become a T cell. And so you can see here are our sort of intermediate positively selected T cells that survive. So you can't be too much, you can't get too little, you got to get just right amount in the thymus. You can see there is also this kind of medium high outcome that I'm not going to talk about today. I'm going to finish talking about some aspects of the Goldilocks model on Friday, and then we will move into our next topic. So I will see you guys on Friday. Sorry I ran a little long.